with us to lecture about the work that he's been doing. And he's interested in and researches the underlying causes of chronic hypoglycemia and diabetes, which the reading that he gave us relates to that. And as he'll explain, normally the body responds to hypoglycemia and will return blood glucose levels to normal levels. But this response is not always there in individuals with diabetes. And in efforts to understand this phenomenon, he has both clinical and animal-based studies to explore and understand this condition. So with that brief introduction, I'm going to turn it over to you, David. Thank you for doing this.
Hello, everybody. I'm Moses. I know one person in the room. So I'm happy to be here today. Really, though, that was my research focus, and I appreciate the introduction. We're going to deviate a little bit from that and not focus so much on diabetes today, but on metabolism, because I think that's the purview of the course, and really focus in on the role of non-neuronal cells in the control of metabolism. So if you think of the brain, most people think of the brain as a collection of neurons, and it's probably the best way to think of it, but what's important to note on a detail level is there's a lot of different cells in the brain that aren't neurons that actually contribute to the control of metabolism. So that's what I want to focus on today. You're going to, I think in the coming weeks, so I'm the first neuroscientist up, I think, and then you're going to have several neuroscientists, so you'll get a lot of background and the brain's control of metabolism and food intake. So I'll touch on that briefly, just enough so that we can discuss the participation of non-neuronal cells in that process. Okay, so my objectives are really primarily, if I can just get you guys some background information, some background knowledge on the different types of non-neuronal cells in the central nervous system, and then I'm going to show just some data to support each of the different cell types, data to support their role in the control of metabolism, and then just as a kind of a tertiary aim is to try to improve maybe your exposure to some research methods that are used to study these cells. Okay, so the lecture is really broken down into about 5 parts. So I'm going to start with the introduction into the CNS control of metabolism. I'm going to use that acronym CNS, which is really just the central nervous system, the brain, and the spinal cord. But mostly when I say CNS, I'm thinking brain. All right, so I'll give you an introduction and introduce the different cell types. And then I'm going to go through the cell types by function. So we're going to talk about how their barrier function, their sensing and signaling, their metabolic and synaptic support, and inflammation of these cells influence metabolism and food intake specifically. All right, so another thing I like to do is, if you see on the margin there, I'm going to break down each of the 5 basic sections into subsections, and you can kind of follow along in that margin where I am. So if your attention drifts, you can always figure out where I am in the lecture and what I'm talking about. And also, I write the title of the slide is going to be pretty much the synopsis of what the information I'm trying to convey on each individual slide. So you can always, again, if you kind of drift, you can always just read the title of the slide. What is this guy talking about right now? That should tell you. All right, so let's just dive right in. So when you think about your brain, often people associate that with thinking or cognition, decision making, right? I decided to come to this lecture, I used my brain to do that, or I'm studying the material. But in fact, your brain is involved with a load of information processing that you're not even aware of, right? It's kind of like your body's autopilot. So it's communicating with all your organ systems and making sure that your body is operating within parameters, and that it basically keeps you alive. But you have very little conscious awareness of that. So when we talk about the brain's control of food intake or metabolism, some of that comes into play where you have this, you don't really have conscious awareness of it, but it is going on in the background, right? There's two areas in the brain that are really important in this, and this is the hypothalamus, which if you imagine a brain, right, a person's head, and you just cut it right down the middle here, so we're looking right in the middle of the brain, right? So the hypothalamus, you know, it's right above the roof of your mouth, basically back there. It's this area right here, and then the brain stem here, medulla, we can call it the medulla, but I usually just say brain stem. These are two areas that are really important in the control of metabolism. All right, so if we think about, again, how the brain is influencing metabolism, one way is there are direct inputs from these areas of the brain out to your organ systems, right? So information, I guess instructions from the brain are going out into your organ systems, and then you're also getting information back from your organ systems that come back to your brain, right? So this is similar to what goes on like at the level of your skin, right? Where you have, you're conscious of this at the level of your skin, where you have sensation, right? So you can feel a touch, you can feel pain or different sensations on your skin, and then the brain adapts to that. So like if it's cold outside, the brain will constrict the blood flow to your limbs, and your fingers will get cold, right? So there's an interaction between the environment and the brain. And similar things happen in your viscera, but you're in your organ system, but you're not aware of that, right? So your brain, so to speak, understands that there's food, is sensing food in your stomach and your intestines. You're not aware of that, but it is sensing it, and it responds. It controls different functions of your metabolism based on that information. All right, to make it even more complicated, the organ systems release different hormones, right, that can, in response to food or different situations, that can influence the brain directly. They can, there's receptors for these hormones in these areas of the brain. So in particular, we'll look at leptin, which is a hormone that's really involved in signaling energy storage. So do you have enough available energy in your body? It's released by the adipose system, so, or the adipose tissue. So that hormone and these other hormones can directly act at the level of the brain, right? And then a third way that I guess your nutritional environment can affect the nervous control of metabolism is nutrients can be detected directly by the brain or by these by the organ system, and then that information is conveyed back to the brain. So really the reason I have this up here is, one, to introduce some of the different levels of control, but I'm also going to show you at least one example of all these different levels, either direct nervous input and from the periphery, and how non-neuronal cells are influenced or are involved in that process. Also, this hormonal mediated change in metabolism, and also how these cells can directly sense changes in nutrients. All right, again, as I said, the brain is often thought of as just a collection of neurons, right? And these cells, usually there's a cell body and then this long process, and you can think of them as information processing units. So they receive inputs here from several different cells, they process that information, and then they send the signal out to another cell. But if you really look at the way the brain is made up, and not just in a cartoon form, what you find is it's much more complicated. And there's not just neurons, but there's all these other cells that are making contacts with neurons, and they can influence this information processing in the brain. So some examples here are the astrocytes, appendable cells, these oligodendrocytes, and microglia. All right, so if we just break down the major cells I'm going to talk about, so 6 different cells, there's 2 different classes. There's glial cells, and then there's this lone non-glial cell I'm going to talk about. It's an endothelial cell, and those are really just the cells that line the capillaries in the brain. They line all capillaries. So the major difference is this distinction, glial cells, and that the root of that word is from the Greek for glue, I believe, right? And what happened is when early anatomists were looking at the brain, it wasn't far off from this cartoon where you had these neurons and all these other cells that seemed to be almost physically supporting them, almost like glue. So it was often thought these cells really had not much function other just to physically support the neurons. But I'm going to present some data that would suggest that they're much more involved than that, than just in that supportive, physical supportive role. So I'm going to go into the different functions. So one thing I want to show you here, so if I list out the 4 functions I'm going to talk about, you'll notice that some cells, particularly like an astrocyte and a tanocyte, they participate in the neuronal control of metabolism through various different functions. So it's not just the cell has one function and that's the function it contributes to metabolism. So it's spread across one cell can have several different functions, and those functions can have several different influences on metabolism. So I'll take it just one at a time, and as I go through, I'll introduce the cell, kind of the background of what it does, and then how that function influences metabolism. So if we talk about barrier functions, so we're going to talk about endothelial cells and astrocytes, and then appendable cells and tanocytes. Right? What's important here is a barrier function. If you can think of the brain in very simplistic terms has almost 2 circulatory systems. So it's perfused from the blood circulatory system, so it's getting blood coming from the heart and nutrients are coming in that way. But there's, and that's here, it's at least diagrammed here, or it's conceptualized there. So there's blood coming into the brain through the capillaries. And there's also this other system of fluid circulation in the ventricles. It's the cerebral spinal fluid, and we'll talk about that. But when I'm talking about barrier functions, it's really that these cells that line the vessels. So if you have the ventricle with cerebral spinal fluid, there's all these support cells, right, that sit, the non-neuronal cells, that form a barrier between that fluid and the brain. And similarly between the blood supply and the brain. So that's what I mean by barrier functions. All right. So, and this is important because these barriers regulate how nutrients enter the brain and also how these hormones enter the brain. All right. So, and that can change based on metabolic state. All right. So if we just start with the capillaries. So a normal capillary out in the periphery that's not in the brain is made up of these endothelial cells here. These are in aquamarine, so this bluish, greenish color, right? And usually they're not very tightly joined. So they have these, there's not tight junctions between the cells. So some of the substances, the metabolites and the protein in the blood can just leak out of the capillary into the tissue. In the brain is very, the brain's environment, I guess microenvironment, the ion concentrations, the metabolite concentrations are very different than that of the blood. So it's a privileged environment. So in the brain there aren't these loose junctions. There's very tight junctions between the epithelial cells, so there's really no passive diffusion in general from the circulatory system into the brain. So these epithelial cells and secondarily astrocytes act as a barrier and also as a conduit for the nutrients to come out of the blood into the brain. So you can see how this works. If this is just let's say a monolayer of epithelial cells, right? There's no real passive diffusion except for lipid-soluble molecules. They can, if they're small enough, they can diffuse across this. But by and large, they act as a barrier for the diffusion of basic metabolites and hormones and large proteins. So the epithelial cells actually have to transport these in various fashions across the what is called the blood-brain barrier. And this can change. This transport can change based on metabolic state. And I'll show you that a little later in this section. But that's just to kind of give you a feel for this idea of a barrier function. There are areas in the brain, so this is another kind of diagram of this where you have a capillary here. These are the epithelial cells in red. Well, that's actually probably smooth muscle, but these are the epithelial cells underneath here. And then the astrocytes form kind of a network around those cells. So it's two layers of cells kind of acting as a conduit between the nutrients there. And this is more of an anatomical view. So this is actual astrocytes filled with fluorescent label. So if you just focus on the red cells, kind of what's going on here is beyond the scope of what I want to talk about. But you can see these cells are sending out these processes here to this, presumably this blood vessel over here. So they kind of send these distal processes out and kind of cover the vasculature. So that's a more realistic anatomical representation of what's going on. Okay, so in certain areas of the brain, those endothelial cells actually are leaky, like they are on the periphery. And these are called circumventricular organs. But what's interesting for this lecture is that a lot of these areas of the brain are right near these areas that are associated with the control of metabolism. So here in the brainstem is one of them called the area postrema, and then the hypothalamus has several different areas where that barrier function of the endothelial cells kind of break down. And what you find if you do histology, if you count the cells in those areas, you actually find there's a lot more astrocytes in those areas. So it does appear, at least circumstantially, that the astrocytes are kind of taking over the barrier function there in those areas. And we'll see a lot later in the lecture, a lot of histology of actual astrocytes and how they're changing in relation to metabolic state. All right, so the other kind of, what I like to think of as like an internal circulatory system is derived from the cerebral spinal fluid. Have people heard of like a spinal fluid or CSF, right? So this is actually a clear fluid that's derived from the blood, and I think it's similar to the lymph system in the systemic in the body. Right? But in the brain, it's this clear fluid, and it really surrounds the whole brain. So it really cushions the brain. Its main, its main purpose is to support the brain. If you can think of the brain, it's sort of like, it has the consistency of Jell-O, right? So if you had this big mass of Jell-O and just set it on a table, it would deform. It would just kind of bow under, right? And that would be very bad for the tissue. It would put a lot of stress on the tissue. So if you surround it in fluid, it's actually neutrally buoyant. So it actually doesn't deform. It just floats basically in your skull in this fluid. So there's the main, people surmise that the main function of the cerebral spinal fluid is as this kind of support function. But it also acts as a sink and a storehouse for a lot of nutrients, for metabolites. It also is a way for kind of intra-brain communication. So one area of the brain could release a hormone, and it can diffuse through this cerebral spinal fluid and reach other areas of the brain. All right, so there has to be a barrier. So there's going to be a barrier then between the brain and the cerebral spinal fluid that facilitates the transport of metabolites and hormones across this circulation system into the brain. And one other thing to point out is that right down here in the hindbrain, the brainstem, and also in the hypothalamus, those two areas of the brain that I'm focusing on, there's two ventricles, which are large kind of fluid-filled spaces. And one is the fourth ventricle down here in the brainstem, and then the third ventricle. And I'll show you some pictures of those, both the cartoon form and the actual anatomy in the brain. So it's really important. What I'm trying to say is those barrier functions for the cerebral spinal fluid are very important in those two areas of the brain, with the ventricles right nearby. Okay, so this is sort of a cartoon of that barrier cell, they're appendable cells, and they usually form a monolayer right here. So this would be the ventricle out here, and then there's this monolayer of cells, and then all the other brain cells inside, right? So there's this barrier between diffusion of all these metabolites and hormones. And this is what it kind of looks like in real, I guess, in a real section. So it's a very thin layer of cells. One cool thing is all these cells have little cilia on them, and if you're looking at really, I do a lot of work in brain slices, so it's really the piece of the brain that I'm looking at is alive, so to speak, at the time. And you can actually see the cilia under the microscope moving. So these cilia are just, they're like little whip-like appendages, and they move that, they circulate that cerebral spinal fluid across the ventricle. So they serve a lot of purposes there. Let's see. So yeah, so this is just to show that they're kind of the guardians for the movement of substances from within the cerebral spinal fluid into the brain. And then they also act to, it's sort of a sink. So if there's a buildup locally of too much waste product or of too much signaling molecule, it can diffuse out into the cerebral spinal fluid, and it kind of protects the brain from localized overconcentration of these things. So now if we look at, okay, so there's a specialized form of these appendable cells. This is the third ventricle I was talking about, and this is a mouse hypothalamus, right? And remember how we were looking at the human brain and it was kind of split down the middle here. In this case, it's a section more like this. So you're actually seeing the right and left side of the brain, the ventricle in the middle, and instead of being very simple sort of columnar cells, these tanocytes, which line the bottom of the third ventricle here at the hypothalamus, they actually send out these processes. So they have these processes that move out into the surrounding brain. So that kind of, they have a little different anatomy than a normal appendable cell, but they still form this barrier. So they still serve this barrier function there. And what's important, right, what I'm trying to convey here is there's these neural circuits. And you're going to get a lot of this in the next 3 or 4 weeks from the other neuroscientists, the specific types of neurons in the hypothalamus that are involved with the control of metabolism, the control of food intake. We'll talk a little later on about POMC neurons, so just remember that kind of neuron. So this circuit is involved in the control of food intake. But what's important here is you have, right next to these cells, these neurons, you have the third ventricle. And you have access to a lot of metabolites and signaling molecules, and these tanocytes are mediating the flow of these signaling molecules and metabolites to these cells, these nearby neurons that are involved in food intake. All right, so how does this change? How does this barrier function? How is this involved in the control of metabolism? Well, it turns out that the tightness of the barrier of tanocytes is actually affected by fasting. So in the fed and the fasted state, it changes. And I don't know if you guys are familiar with immunohistochemistry, so staining tissue with fluorescent markers for a molecule. But in this case, the green, you see this green labeling around these tanocytes, that's really a marker of, they're just tagging a protein, and it's a marker for how well these cells are joined together, how tight the junctions are between the cells. So it's just analogous to how permeable or tight the diffusion is. And what you find is, like in a fed state here, if we just say the green color is how more green, more barrier, less green, less barrier. Right? So there's not so much green here in a fed state, but just a 24-hour fast. I believe just in a 24-hour fast, you see this large increase in that barrier function. So the barrier gets much tighter there at the ventricle. So it at least appears that these cells are responding directly to metabolic state. So the animal, so it's not just, what I'm trying to get across to you guys is when you're hearing the lectures in the future, which is very important, all the nervous control. But just keep in the back of your mind that there's also other cells in the brain that are responding to metabolic state, and that it's not one or the other. It's the synergy between all those cells that really control behavior. All right, so that's one example of how the barrier function of these cells is changed by metabolism. So next I want to talk about sensing and signaling. And really what I'm trying to get at here is, if you remember, the tanocytes, both the tanocytes and the astrocytes had these long processes that seem to join their cell bodies out to these either the circulation, the blood vessels, or the cerebral spinal fluid. And there's some evidence that there is a signal transfer. So even though there could be a signaling molecule in the ventricle, right, that doesn't necessarily diffuse into the hypothalamus, it could activate a tanocyte, which could convey that information into the brain to an adjacent neuron. And the same thing would be analogous from the blood supply into the brain. So it doesn't necessarily mean that there is a transfer of the signaling molecule. There actually could be just, these cells could be act as sort of sentinels for that signaling molecules without it actually diffusing into the brain. All right, and I'm just showing that, right? It would be, you know, these cells are almost sending out these processes that can sense something here, and then that could be conveyed to this cell body. And if you look at all this cloudy, kind of red cloudy, that's all the connection that cell has to other cells. So you can imagine a situation where this cell could sense what's going on in the blood and convey that to cells that don't have that close opposition to the blood supply. All right, same thing with tanocytes. Remember, they have these processes that go out into the hypothalamus. Again, could convey a signal that way, and that signal could be conveyed to these underlying neural circuits that control food intake. All right, so how do we measure that signal? So how can we get an idea if a cell, a non-neuronal cell, is activated by something and how it may signal? Oh, sorry, I got ahead of myself. So this is an example in the brainstem. This is a tanocyte. This is actually from some section I took, some staining I did. You can't see it too well, but this is a stain for glial cells, this GFAP, and this is the ventricle up here. You can see it a little better over here. So you see, again, these green cells are neurons. So you see that this cell can sense stuff out in the ventricle and convey it into these cells that don't have access to that. So again, the question is, going back to the original question, which was, how do these cells get activated, right? What's going on? How can this cell get activated here and convey that information into the neurons? So one thing is through calcium signaling. So it turns out that these cells possess the machinery to really be robust calcium signalers, all right? So they respond to a variety of different metabolites and signaling molecules. So I'm just going to show you a video that shows in a slice, and you can see these cells flashing, right? So what that is they're oscillating. Their intracellular calcium levels are oscillating, and in response to a substance we put on this slice, right? So you can see this dynamic signaling. You can imagine that if they have access out into the brain to some of the signaling molecules, that could be conveyed back into the soma. They could get it activated, and then that signal can be conveyed to adjacent cells. All right, so I just wanted to give you a little background on how we would do that. So this is now a rat brain. All right, so it's very similar to that section to the human brain. It's right down the middle of the rat's head, right? And this is the brainstem, the area of the brain that I'm more interested in. So what we can do is, in an intact animal, while they're under anesthesia, we can actually inject dyes into this part of the brain. So one is that green signal you saw flashing. Of course, I spelled it wrong. I have to fix that eventually. And then this other substance, which is sulforhodamine 101. And this is a stain that's only picked up by non-neuronal cells. So this allows us to differentiate between a neuron and a non-neuronal cell. And I'll show you what this looks like. So we inject this, and then this is the area of the brain we're interested in, and what we end up doing is making these thin slices right through that area of the brain, right? And they look like this. They're not colored like this. It just looks like brain tissue. But this right here is the area of the brain. It's called the nucleus of the solitary tract. That's the area of the brain that we were interested in this study. And you can zoom in on that area of the brain and look at the cells, and what you end up seeing is this green signal. And what the N and the A mean in this is an astrocyte, which is a type of non-neuronal cell. N is neurons. So the green, that calcium signal sensor, is picked up by both types of cells, but that red, what we're pseudocalling red here, this SR101, is only picked up by the astrocytes. So when you look under the microscope and you do an exposure of this tissue, you can actually pick out individual astrocytes as opposed to the neurons. And you can look how the different cell types respond to different stimuli. All right, so this is some work I did several years ago, but this was just to look at these brainstem astrocytes and see if they responded to changes in glucose concentration. So we're going to go from a 5 millimolar glucose solution down to a 0.5 millimolar and then return to 0.5 millimolar and then return to 5 millimolar. And what I want you to do is kind of follow. I'll show a movie over time. And if you'll follow these astrocytes, you'll see there's also going to be a slider that moves across here. So you'll see when you enter the low glucose as opposed to returning. And these cells brighten up and then kind of dim back down. Let's see if this will work, right? So if you look at them, see how they're brightening up here, and then as we return back, they kind of dim back down. I think it runs twice. So they brighten up. So there seems to be a signal that's conveyed that returns back to normal after the restoration of the normal glucose. And you can actually plot this as a signal intensity over time. So you can, basically you're going to just circle this cells, you're going to put a region of interest around the cell and track its intensity over time. And you can graph this up here over minutes, right? And you can see if this is just kind of relative intensity. So how much calcium signaling is going on. So there's this baseline level. It intensifies during the low glucose, and then it returns back to normal following the restoration of the normal glucose. So these cells actually signaling in response to a change in glucose availability. So a change in nutrient availability. So there's also evidence that tanocytes can do this. So this is not my work, but some very interesting work. There's no movie here because it just came from a journal. But the idea is it's a much lower resolution. So just imagine this to be a collection of tanocytes, right? This is a low resolution. But this is the ventricle here, and the tanocytes lining the ventricle. And there's a little micropipette, very small little glass, I guess you would call it, in this case it's almost like a little glass syringe, and it can puff out a glucose solution. So in this case I think they go to a high glucose solution, so they can puff on high concentrations of glucose there, and you can see as they puff it on, the red is an increase in activation. So you see these cells are activated by that transient change in glucose availability in the CNS, and then it kind of dies down to normal over the half a minute or so. And you can imagine, right, that even though that glucose may not be quickly being transported across into the brain and changing the neurophysiology in that way, but these cells are sending all those processes into the brain so that they can sense and pass that information on to adjacent neurons. All right, so I think that's the end of that section, right. So again, you guys stop me if there's any questions. I'm happy to discuss. It's going to be a lot of information, but I'm just really trying to give you a feel that these cells can participate in the control of metabolism in addition to neurons. All right, so let's talk about metabolic and synaptic support. So the first part of this really gets interesting. So we've already talked about kind of how maybe some of these cells could sense and signal changes in nutrient availability. But their physiology is actually changed by what you eat. right? So just your diet can change their physiology, which can affect the brain as a whole. So let me show you how this works. So what I'm really saying is, it's not only nutrients being detected directly by the brain, but also you're ingesting nutrients, right? And what you ingest actually changes the physiology of the brain. And something that's important is that these glial cells actually are metabolically linked with neurons. So if you think about a metabolic unit in the brain, it's really a collection of two cells. It's the astrocyte or the tanocyte in some cases and the neurons. There's this active transport of metabolites between the two cells and amino acids between the two cells. And what's really important here is that the brain actually uses amino acid as signaling molecules, the major neurotransmitters in the brain. So that's the chemicals that nerve cells use to communicate with each other. The two major types of neurotransmitters are amino acids, glutamate and GABA, right? So you're actually using metabolites, the brain uses metabolites to signal. So you can understand how if you change your diet, it may change the availability of these amino acids. So it turns out that the TCA cycle of glial cells, which you could think of as astrocytes and tanocytes, they're actually linked with the TCA cycles of neurons. So there's really, the cycle is really not independent. There's this transfer, this shunting of metabolites over from the glial TCA cycle to the neuronal. And in fact, if you think, most people, I think it's generally right that the TCA cycle is about generating energy, right? You're oxidizing metabolites to generate energy. In glial cells, they're actually deriving most of their energy from glycolysis, and they're using the TCA cycle as a synthesizing cycle to synthesize these signaling molecules. So there's this interplay between these signaling molecules between glial cells and neurons. And you don't have to understand this, but it actually gets more complicated. But what I want to point out is that these major signaling molecules, glutamate and GABA, there's this interplay between the astrocytes and the neurons in the production of these two. And if, just for our purposes, we can think of glutamate as kind of the excitatory. So if you think of a volume control, more glutamate, you get more activity in the brain. More GABA, you get less activity in the brain, right? So a healthy brain kind of has a good balance of these two neurotransmitters. Diet can actually influence the balance of those two neurotransmitters. And I don't know if you guys know about a ketogenic diet in the treatment of epilepsy, but it's been known since like ancient Greek times that diet high in fat and protein and low in carbohydrates actually is an effective treatment for epilepsy. So epilepsy really can be thought of, right, people with epilepsies have seizures, but that's really, the underlying cause is there's way too much glutamate, so the brain is overexcited. So you can actually use diet to change the relative amounts of the neurotransmitters. So it's thought that that's the way the ketogenic diet works, right? If we go back to this, instead of metabolizing mostly glucose, then you're metabolizing mostly fat and protein, and it ends up that the glial cells actually change how much of these different signaling compounds they're generating from those substrates, and you get an increase in GABA and a decrease in glutamate, which leads to just an overall decrease in seizure activity. So as you can see there, there's a kind of a subtle interplay between neurons and these non-neuronal cells and the way even just the food you eat affects your brain's function. All right, so there's another way that astrocytes are involved in signaling, and it's really kind of in support of neuronal signaling. So remember, there's this facet of the neuronal control or the brain's control of metabolism where there's actually signals being sent out to the internal organs, and the internal organs are sending information, so to speak, back to the brain, right? through these cranial nerves. And one of the most important cranial nerve, the vagus nerve, comes into the brainstem in that same region that I showed you before in those slices that I was studying the low glucose response of astrocytes. But it turns out that there's a lot of information coming in by way of this nerve. And just in case you guys didn't know this, right? So nerves, if you think of two neurons communicating, right? There's if you think of this as the nerve that's communicating from the organs up into the brain, right where the two cells meet, there's a release of signaling compounds like this glutamate and the GABA that I talked about, right? And so this is the way the brain, just kind of the traditional way that neuroscientists generally think of brain signaling, is this release of signaling molecules between this presynaptic neuron and a postsynaptic neuron. In our case, let's think of this as the neuron coming from, say, the stomach, and this is the neuron that's in the brain, right, that's receiving that information. But if you look at any synapse in the brain, like realistically under an electron microscope, what you find is that around every junction between a presynaptic and a postsynaptic neuron, there's this astrocyte process, right? And it's been pretty conventionally showed that the information transfer between these two cells is influenced by this process. Okay, so if you think about this in a cartoon, so here's the information coming from the stomach, right? This is the neuron that's in the brainstem, right? And generally there's this release of signaling compound from this neuron to this, right? But the signaling compound can actually interact with this astrocytic process that is around the synapse, and that can lead to activation, this calcium, increase in calcium signaling in the astrocyte, and then the astrocyte in turn can release signaling compounds onto either the presynaptic or the postsynaptic neuron, and in that way it just changes the way the information flows between the two cells. So if you're getting information, say, from your stomach about the presence of food, right, so the astrocyte is involved in changing the gain of that signal through that information transfer. And we actually can use this same technique to actually examine how the astrocytes are involved in this, how they're involved in this modulation of signal transfer between neurons from the periphery into the brain. So we can label the cells, but then if you cut them on a different axis, a different plane, right, it's kind of on the plane here, a horizontal plane, you can actually preserve some of the the terminals from the nerves coming from the stomach, right, and I'll show you what this looks like. So here's our area of interest. This is the nucleus of the solitary tract. Right, so that's going to have that postsynaptic neuron that's receiving information from the stomach, so to speak, right? And then right here is called the solitary tract. So this is all these nerves that are communicating information from the stomach and the other organs. They send their little processes right in this solitary tract, and it kind of curves around and interacts with these cells here. And what we can do is put a stimulating electrode right there on that solitary tract. Now, of course, those processes are all cut, right? So it's not an intact circuit, but if we put a stimulating electrode there, we can mimic the way those cells would actually communicate with the brain. So we can dial up the stimulus protocol so we can really correctly mimic signals coming in. And then we can look over here, we can image over here and see how those stimulating those fibers actually changes signaling in the astrocytes. So I'll show you, again, we have the kind of orangish looking cells and the green cells of the neurons. And what I'm going to do, I'll play a video, and it's 3 stimulus trains one right after the other. So you'll just see this flash, flash, flash. And all the cells really respond very robustly to this. But if you kind of concentrate on these, you can see that they are, if you kind of ignore the rest of the flashing, you can see that they kind of come up as well. But you see that flash, just like Christmas tree lights. So there'll be 3 of them, and then it'll loop back around. So you're actually seeing like living brain tissue respond to this stimulus we're kind of stimulating, we're simulating what it would be like to have information coming from the periphery. All right, so again, we can just concentrate on these cells, and we can blot this change in fluorescence in response to these stimulus trains, and you see that there's this nice quick transient increase in calcium signaling in response. So that was novel just by itself, that there was actually this going on, that this information from the viscera were actually activating these astrocytes here in the nucleus of the solitary tract. But we actually took this a little further, and I'm going to try to explain the experimental design. Basically, we wanted to know what were the signaling molecules, what were the receptors that were involved. So we know that those fibers that come from the periphery, they release glutamate. So we just wanted to know what kind of glutamate receptor was on the astrocyte that was mediating the signal we were seeing, that flashing. How did we get to the flashing? So one thing we can do is we'll do an electric stimulation and then wait 10 minutes and then stimulate again. Right? And if we do nothing in the intervening time, what we find is that if you measure in the same set of cells, right, the same slice, basically you get the same effect after 10 minutes. You just wait 10 minutes, let it rest, and it'll give you the same effect. In subsequent experiments, we apply different drugs to see if we couldn't block a certain kind of receptor and see if we couldn't make this second stimulation go down, right, if we couldn't block it. So we tried different receptors. One was a metabotropic glutamate receptor, and this certain drug blocks that specific kind, and there was no effect. There was another kind of metabotropic glutamate receptor we tried, no effect. NMDA receptors. These were all the common receptors that have been shown in other areas of the brain mediated this response. So we were using the likely candidates, but what we found was it was a very unlikely type of receptor. It's an AMPA receptor, which is, it's not uncommon at all. It's one of the most common types of glutamate receptors in the brain, but it's usually on neurons, not on astrocytes. But you can see when we put on this drug, there's this nice decrement in the response to the second stimulation. And we actually showed that you could just put on AMPA, you didn't need to stimulate or anything, if you just put on AMPA on a slice, that it would light up all the astrocytes. And AMPA just selectively activates this kind of receptor. So really why we do these kind of experiments is, it wouldn't be that easy in practice, but the theory behind it is if we can figure out what kind of receptors involved, then you can design a drug that would target just that response, just the astrocytic response and not the neuronal response. It turns out it wouldn't be that easy in this case, but it's still useful information in the future. So that's just showing how astrocytes can participate in, they do get information from the organ system about what's going on in the microenvironments in the organ system there. Okay, so if we come back to this diagram, and remember there's this astrocytic process that's going around all these neurons that are communicating with each other. And what other groups have found is that there's a big influence not only on diet, but on hormone levels, and I'll show you leptin in particular. They have a big influence on the size of this process, and then an underlying influence on the information flow in the synapses. So a lot of it is kind of circumstantial, right? I can put on leptin and it'll show, I'll show you some histology, and you treat the animal with leptin and the astrocytic processes get it smaller, right? Or they get larger. And then that leads, you know, if we then measure food intake is also affected, but you don't really know if it's because of the astrocyte or because of the leptin itself. So I'll show you some circumstantial evidence that'll gradually get more specific. All right, so this is one. So if we just look at the green here, that's that astrocytic process that's going around a palm C neuron. And if you treat this animal, or you just put it on a high-fat diet, just giving it a different diet changes the amount of coverage of these astrocytic processes, right? So that's just one section, but if you count them all up, I didn't, but if these guys count them all up, you see this nice increase in the amount of coverage of these processes around these neurons with a high-fat diet. Okay, and of course it leads to a change. So this is the chow diet, and this is the high-fat diet. And when they look at the response of neurons in a field to changes in glucose, there is this change, right? But again, what does that mean just because the astrocyte changed, that you have a change in neuron? It's hard to say, right? It's hard to say in that case, but it's like circumstantial evidence that there is an effect there. One thing that is interesting is leptin. Leptin receptors or astrocytes do express leptin receptors, and you can change kind of the size of the astrocytic projections or these processes by just giving leptin. So this is a control animal just stained. I believe this is just stained for an astrocyte stain. It's not specifically leptin. But if you give leptin, they tend to ramify more. They get more processes. So if you go through and you measure every process here, what you find is that the length of each projection goes up when you treat with leptin. All right, but more concrete, I guess more concrete evidence comes from this study. And it's really hard to see, I understand that. But if you just look up here, all right, so what they're trying to show is you stain now with IHC for astrocytes with this GFAP, and then you stain for leptin receptor. You can see some evidence, though it's not obvious probably from the back of the room, but there is some evidence that there are actually leptin receptors on the astrocytes. Okay, so what you can do then is you can generate a leptin receptor specific knockout mouse that will just take leptin receptors out of astrocytes only and leave leptin unchanged in other cell types. So that's more of a conclusive experiment. Can we just modulate astrocyte leptin signaling and have an effect on behavior or metabolism? And this group indeed showed that. And this is again in the hypothalamus around these POMC neurons, this type of neuron that's really important for food intake. And what you find, this is the blue is a wild type animal, and these red bars are the knockout animals. So that's the animal that doesn't have leptin receptors, just in astrocytes. And this is measures really of the number of synapses, so the number of connections between these neurons. So when you, just by knocking leptin receptors out of an astrocyte, you actually affect how many synapses the neurons have, so how well they're communicating with other neurons, and also their activity. If you think of these little dashes as activity, so the more dashes or the more downward deviations, the more active a neuron is. So in general, if you record from a bunch of neurons, what you would find is by knocking out the leptin receptor in a whole other different kind of cell, right, on an astrocyte, you actually lead to more activity in these POMC neurons. And really the most important part of this is that it has an effect on behavior. So it actually changes the animal's food intake. So here's the amount of food these mice eat over the course of several days after, I believe it was a conditional knockout, so they were able to administer some compound and acutely knock out leptin out of astrocytes, and you find that over time there's this deviation so that the animals without leptin receptors in their astrocytes ate more food than the control animals. So there seems to be a pretty definitive link between leptin signaling in astrocytes and the control of food intake. So this is more, I would say, again, circumstantial evidence, but it's a different part of the process, right? We were talking about how the astrocyte was influencing how two nerves talk to each other, and we were focusing down here on this end where the information was coming into the neuron and affecting it. There's another end of the system here where the neuron has received the information, but now it's transmitting it on to another part of the brain. And there's these glial cells, they're called oligodendrocytes, and they actually wrap myelin, which you can just think of like an insulation around the nerves. Right, so the more insulation you would say around a nerve, the faster it conducts and more efficiently it conducts information. So you can actually change, acutely change how these neurons in the hypothalamus are insulated, so how well they communicate downstream by you put on, not not us, but this group put on this compound. It's called MPY. It's neuropeptide Y. It's a neuropeptide. It's not a hormone, but it's acting like a hormone. It's very important in the control of food intake. But if you just put on MPY, you can actually, so if you can think of that myelin as being wrapped around these axons here, or this neuron here, just putting on MPY increases, you can see how it got much denser. And that the supposition there is that you've changed how well these neurons communicate their signal to other areas of the brain, right? And this is graphed here, so there's, about a 5% increase in the amount of myelin just in response to this signaling molecule that's really involved in food intake. It's not implicated in any way with some kind of astrocyte physiology. So it's really specific, I think, for the hypothalamus. So again, just some evidence that non-neuronal cells are responding to these compounds that are very important in the control of food intake. All right, last and not least, inflammation. And I'm not an inflammation guy, so this section is pretty short. But again, there's two major cell classes that are involved in inflammation. Primarily is the microglial cell. All right, if you followed along in any way, you'll notice that I haven't even talked about microglia. Right, they're only really involved in this inflammation process. They do a lot in the brain, right? But they are basically immune cells. They're resident macrophages, right? And they serve like other immune cells in the body. They respond to tissue damage. They phagocytize cellular debris. They also release growth factors and help in brain development. They in turn, when there's injury to the brain, they can release growth factors that aid in the wound healing and things like this. Right? So they serve this beneficial function in the brain. But if they get, like other immune cells, if they get constitutively activated, so if they're just always activated and always kind of in this reactive state, this can move from being a beneficial effect to actually damaging the neurons. So really, the take-home message is when microglia get activated for a prolonged period of time, it's not good for the underlying tissue, the neuronal tissue, the neurons. And one way that that's measured is this compound called IBA1. Stands for ionized calcium binding adapter molecule. But for our purposes, really all we need to know is, if you think of this as kind of a non-reactive microglia, here doesn't express much IBA1, but as they become more and more activated, they become more, what are we talking about? If they get more reactive is the verb I'm looking for. So as they become more reactive, they express more and more IBA1. So it's a common compound that's assayed to see how reactive microglia are. Okay. The take-home message, though, is this slide here. So again, this is the hypothalamus. So this is the third ventricle again. This is where all this neuronal circuitry involved in food intake is. And all they've done, this group, is stain for IBA1 just in a standard chow diet. And then you just put the animal on a high-fat diet. I think it's for like a week, I forget. It's a really short period of time, and you see that all these microglia get reactive. They start expressing this IBA1, and subsequent papers have really shown, this is a big body of work in the hypothalamus now, that chronic exposure to a high-fat diet can activate these microglia, and it leads to damage to damage of the neurons in the hypothalamus. So it was again, very interesting way that just diet can affect some of these non-neuronal cells and lead to changes in the underlying neuronal circuitry. It's not just microglia. If you notice down here, astrocytes can also become activated. They're not an immune cell, but there is some evidence that astrocytes do participate in wound healing in the brain, and also clearing debris, cellular debris. And when they become overactive, it can lead to kind of negative consequences. So again, this is not as well fleshed out, but there is some evidence. So in this case, instead of EVA1, we look at vimentin as a marker of astrocyte activation or inflammation, and you see that if you treat an animal with leptin, you get an increase in this, in the fibers or the astrocytic processes that are expressing vimentin. So it appears that leptin can lead to activation of astrocytes. I don't think it's all that clear whether this has any kind of physiological consequence or if it's pathophysiological, right? This could just be a normal consequence of a high-fat diet that just, or exposure to leptin. So it doesn't necessarily mean that anything is going wrong here, is affecting the neurons at this point. But it is interesting to see that, given what we know, or what I presented, that astrocytes have an influence on neuronal behavior, that just giving them leptin can change their state, so to speak, to being more activated. All right, so that's it. So I welcome any questions.
You mentioned about the ketogenic diet for epilepsy and the neurotransmitters.
Mm-hmm.
Are those, the GABA and the glutamate, are those the same neurotransmitters that are responsible for kids that have ADHD? And if so, would a ketogenic diet help them?
I don't think so. The reason why, no, I'm not a clinician, so I'm just kind of pulling from my, just what I know anecdotally. But I know that the way children, or even any individual that has ADHD is treated is by giving them stimulants, which seems kind of counterintuitive. So I think it's actually the opposite, right? So you wouldn't want less, you wouldn't want more gabatone. You would actually, it's something about, I don't know, I kind of conceptualize it as, instead of, there's not enough activity, so the brain is thinking about a ton of things at once. Whereas if you stimulate, so you're probably increasing glutamatergic tone, so it's the opposite of a ketogenic diet. It would actually cause the brain to kind of, it allows the brain to function, focus in on certain tasks. That's all cortical things. It's kind of outside of my area of expertise. But I do know that it would be opposite. Like I know you prescribe stimulants for ADHD, so that would be opposite of the effect you would expect from a ketogenic diet. So you would actually predict that it would get worse if given a ketogenic diet, but I don't know. More questions? Sure.
Do you know if there have been any studies that look at non-neuronal cellular activity in bariatric patients?
That's a good question. I'm sure there has been. So you mean like in bariatric surgery?
Mm-hmm.
Yeah. That's interesting. There's a lot of evidence that, I'm looking at Crystal because she might tell me I'm wrong. No, I don't know a lot about, and I don't think Crystal knows a lot about bariatric surgery, but she may know more than me. There's some evidence that there are some signals that are coming from the organ system that's mediating some of the effects. So it's possible, but whether, and I think it's likely somebody's looked at it, but don't know the specifics. But it seems very likely. But again, you just want to think of it as a synthesis. Like it's not just the neurons, it's not just the astrocytes or the glial cells. It's kind of how they're working together. So yeah, it's very possible. I think that's very possible. Something that's going on.
I don't know if Hans Rudy's gonna.
Oh yeah, that would be, yeah,
So
he'll be
when
in a couple weeks.
You might want to ask him that question.
But he's, yeah.
He's the bariatric surgeon.
He doesn't know much about glial cells. All right.
What?
Okay, well, he doesn't appreciate it. No, he doesn't
Say
appreciate it.
that.
So, but you can give it a try. Other question?
So I have a question. I think you said it was the astrocyte that sort of cleans up the garbage there.
Yeah, that and the
So-
microglia, yeah.
And macrophages in the blood do that, but they'll do it until they actually burst, and then you
Right.
have an oxidative kind of
Yeah,
condition there. Does that happen?
It's very possible. Again, that's kind of outside of my area of expertise, but there is this, where is it? Where, this slide, where you get, you know, that sort of reminds me of the reactive oxygen species and stuff. So it
Right.
could be that they're actually rupturing and and all those compounds that they're using to digest that cellular debris is then being liberated out into the tissue. So yeah, it's not a good scene, that's for sure.
Yeah.
But I don't know the specifics of the mechanism for how that happens. But that's a good question. Yeah.
I'm just thinking that in that case, in terms of nutrition, those
Yeah.
vitamins and molecules that help control
Right.
that would be
Right,
would
right,
be
like
an advantage there.
antioxidants and things, yeah. And that's, there's a lot of evidence in neurodegenerative diseases, you know, the Parkinson's and Alzheimer's, that some of the mechanism of action of some of the sort of antioxidants and are acting at the level of glial cells,
Right.
yeah,
Okay,
to be
interesting.
neuroprotective. Yeah, so that's another good point. Sure,
I don't know if this is related, but
we'll give it a whirl.
so there is some people when, like, if we are talking about glucose homeostasis, some people get hungry faster than other people.
Okay.
So what is the difference when all those signals?
Yeah, again, that's going to be a better question in the coming weeks for Dr. Morrison, Dr. Butu, and Dr. Munsberg. But so the mechanism of why people would become hungry faster, is that the idea?
Yes, because it's like some people just like they eat and have you showing there's some signals that
Yeah.
activated the brain, but some people can be without eat like
Right.
forever and they're fine.
Right. Yeah. There's certainly, I know there are genetic conditions, you know, where you have like a leptin receptor deficiency or something like that. But also there could just be, I think, there's even what's called leptin resistance, I think, that may be going on there. So even though there's, so your adipose tissue, when you have plenty of nutrients, right, you're well fed on a long-term basis, say you've just overeaten for days, you know, for a week, there's a lot of leptin. And leptin should normally reduce your food intake. But there's good evidence, I think, in humans and in animals of leptin resistance, which means, you know, when you're in chronically, when you have chronically high leptin levels, that the brain stops responding to that. So it's kind of a homeostatic mechanism where you have so much of these satiety hormones in your circulation that the brain kind of stops responding to them. It's like an overstimulus. So I think that could be going on in that situation where the normal signals that would keep one individual from continuing to eat no longer are effective in the brain for whatever reason. And usually it's some kind of resistance. I mean, it's very common in the nervous system. Think of it this way. Have you ever gone, I think everybody's probably experienced this, if you go swimming early in the year and the water's cold, right, and you fight to get in the pool or in the ocean because it's so cold, and then it's just like excruciating. But then after 10 or 15 minutes you feel fine, right? It's like it doesn't feel cold anymore. So the nervous system is often changing its game so that it's sensitive to changes, right? So it would be kind of analogous to that, like a person who has really good, you know, that doesn't have high levels of leptin can sense really small changes. It can sense that cold water, right, when they get into it. But if you've had high levels of leptin over a long period of time, so you've adapted to the cold water, you no longer sense it being, you know, what your body's really sensitive to, or the nervous system, is a change over and above that. and you're not gonna produce that. You just kind of topped out the leptin. So I think that may be what's going on, sure. But great question for Dr. Morrison next week, and even Crystal after this. Okay, go ahead.
Yeah, I just asking, is it possible that there is a limited number of the leptin receptors? So like if like people like got gain the weight and the leptin actually is increased, but the number of the leptin receptors is in the brain is like has some,
Reduced.
not reduced, maybe keep the same, but it's like saturated.
Yeah.
No matter how much like
Right.
leptin you have now, but
Yeah, I think that, I don't know the exact mechanism of leptin resistance. I know it's been studied a lot, so I can think of that's a likely mechanism, is that you can saturate a receptor. You just, and then often when you're saturating a receptor, you get this adaptive process where the receptor is not as responsive for whatever reason to the signal. So, yeah, that's certainly a possibility. Yeah, and there's so many different hormones too. It's not just leptin; it's CCK. What are some of the other yeah, there's all those. Ghrelin's like the fasting hormone, right? So it would be a drop in ghrelin. So there's push-pull systems too. Yeah, I'm not as well versed, but you'll be getting a lot of that in the coming weeks. It's very interesting, so I like it.
Other questions, comments? Thank you.
Sure.
Very interesting.
Thanks.